welcome to this webinar on improving the credibility of your study when there's missing data. So here's what we'll cover in the webinar. We'll have an introduction covering what is missing data and why it's important, with a simple example from an illustrative data from Parkinson's disease. Jessica Cooper will talk about preventing missing data, and we have 3 case studies there. I'll talk about the third one because it's about how the statistician can work to help prevent missing data. Ilya Lipkovich will then talk about multiple imputation as a good framework to deal with missingness, and he'll have an example of sensitivity analysis for time to event outcome, which is a relatively new area for missing data research. Finally, I'll talk very briefly about how to have an evidence-based strategy for your missing data, and to discuss that, I'll use an example from major depressive disorder. Then Jessica will give a summary. So for purposes of this webinar, missing data in a clinical trial are data that were planned to be recorded but are not available. That could be due to missed visits, perhaps clashing appointments, but the major source of missing data is early withdrawal of subjects. Now, as we know, usually clinical interest is in symptoms at some defined follow-up time, often the planned last time point of the trial. And the question that people are beginning to ask more earnestly is, can the results of a trial retain credibility if we don't know the symptoms at the planned time point for a proportion of subjects whose data are missing? For all we know, all the withdrawals in the experimental arm could have had an unrecorded devastating side effect. So up to recently, a rather restricted number of approaches have been taken to missing data. Sometimes missing data is simply ignored. That's called complete cases approach or available data approach. A common solution, in quotation marks, has been to use the latest available observation, often called last observation carried forward, or LOCF for short. And that's been justified as probably conservative because the subject is likely to have had their poorest symptoms just prior to dropout. But there is growing concern among everyone, among regulators, statisticians, clinicians, and patients, because with these methods, it's really been difficult to assess the credibility of trials with missing data in any evidence-based or scientific way. There's lots of evidence about this growing concern. One that people quote a lot is the case of Xarelto, where a submission was rejected. The panelists, among other things, said it was not acceptable not to know whether 9% of patients were alive or dead, and, quote, the missing data in this is troubling and concerning, says another panelist. That's just one example, though, of the growing concern. Now let's look at what missing data looks like, if you like. We don't see it, of course. But this example is from an illustrative data set in Parkinson's disease, which features in a book which Ilya and myself and others contributed to, called Clinical Trials for Missing Data, which Andrew mentioned. So here's simply a plot of the mean efficacy scores in an illustrative Parkinson's disease data set. The vertical axis is an efficacy score, perhaps the UPDRS or a subscore, and the horizontal axis is time in weeks. And you can see that the control arm, represented by the solid line, has efficacy scores that are clearly worse in the mean than the experimental arm, because lower scores are better. The experimental arm is represented by dashed lines. However, these plots, since they are plain, simple means of the scores, ignore subjects who withdrew. Sure, the experimental treatment looks much better than control, but if we look at a plot of withdrawal from the study, the technical description is a plot of the probability of staying in the study. Here again, the solid line represents control, dashed line represents the experimental arm, and again the horizontal axis is time in weeks. And you can see that from the very start of the study, the experimental arm subjects, represented by the dashed line, are withdrawing at a more rapid rate than the patients in the control arm.